Hello and welcome to the course Basics of Experimental Design for Cognitive Psychology. I am Dr. Ark Verma from the Department of Cognitive Science at IIT Kanpur. This is week 8 of the course and we are talking about event-related potentials. In this lecture, we will go through go through an overview of the basic steps that you need to put together an ERP experiment. We have seen through examples how are ERPs useful to study mental processes, how are they useful to underline and basically carve out specific aspects of temporal processing in the brain with respect to particular events or stimulus. In this lecture, we will basically look at how an ERP experiment is basically structured. Now, the first step is setting up the recording of the EEG. So, the EEG is typically recorded from electrodes on the scalp with a conducive gel or liquid between each electrode and the scalp, so that there is maximum conductivity and it basically makes for a stable electrical connection. The electrical potential or the voltage can then be recorded from each electrode, resulting in a separate waveform from each electrode site. This waveform will be a mixture of actual brain activity, biological and electrical potentials produced from the outside of the brain by the skin, eyes, muscles, etc., and induced electrical activity from the external electrical devices that are also picked up by the head, the electrodes and the electrode wire. So, this is actually a lot of noise that you capture from each electrode site. If precautions are taken to minimize these non-neural potentials, the voltages produced by the brain, that is the EEG, will be relatively large compared to the non-neural or noise voltages. So, one of the first and the more important things is to basically minimize all kinds of noise that can be captured here and to maximize the neural activity or the the EEG that is arising because of the neural activity of the brain. Now, typically this EEG signal is relatively small, under 10 millivolts, 10 microvolts, sorry. So, the signal from each electrode is usually amplified by a factor of 1000 to 10,000 times. The amplification factor is called the gain of the amplifier. Previous experiments, if you remember the oddball experiment that we talked about where we were working with the P3 wave, it used a gain around 20,000, and the next experiment that we were talking about, the one with schizophrenic patients, used a gain around 5,000. So, that is basically by the factor of which you multiply the basic signal. The continuous voltage signal is then turned into a series of discrete digital values for storing on a computer. In most experiments, the voltage is sampled from each channel at the rate of between 200 and 1000 evenly spaced samples per second, which is basically 200 to 1000 hertz. The EEG is typically recorded from multiple electrode sites across the scalp, and there are different studies which have different preferences. For example, some studies use only around 5 to 6 electrodes, whereas some studies may use up to 256 electrodes, depending upon the quality of the data that can be obtained, or the research question, the areas of the brain, and the kind of processing that you are actually looking for. Now, after the setup of the recording the EEGs is created, the next very important step is the artifact rejection and correction. The raw EEG recordings, as we were just talking about, pick up several common artifacts which require special treatment, which requires us to handle them. The most common of these artifacts arise from eyes. When the eyes blink, a large voltage deflection is observed over much of the head, the entire scalp, and this artifact is usually much larger than the ambient ERP signals that you are actually interested in. Sometimes eye blinks are systematically triggered by tasks and may vary across groups and conditions, yielding to a systematic distortion of data, which again can be interesting for the kind of question that you are asking. Large potentials are also produced by eye movements when the eyes move, and these potentials can confound experiments that are interested in presenting lateralized stimuli, or they focus on lateralized ERP responses. So, in all of these lateralization studies, you want to have minimal eye movement and minimal artifacts based on eye movements. So, trials containing blinks or eye movements or other artifacts are typically excluded from the averaged ERP waveform. This approach, however, has two shortcomings. First, a fairly large number of trials will need to be rejected because they contain these artifacts, and it reduces the number of trials contributing to the average ERP waveform. Secondly, the mental effort involved when you instruct your participants to not blink their eyes, the mental effort involved in suppressing the eye blinks may also sometimes play in, and it may impair the task performance of a participant when you are asking them to do a given task. These problems are especially acute in individuals with neurological or psychiatric problems who may blink on almost every trial or may perform the task poorly because of the effort that they devoted to blink suppression. So it's a basic trade-off that ERP researchers have to be extremely careful about. Fortunately, methods have been developed to estimate the artifactual activity and subtract it out, clean the data, leaving artifact-free EEG data that can be included in the average ERP waveform. The third and a very important step is filtering. Filters are usually used to remove very slow voltage changes which are under 0.01 to 0.1 Hertz, and very fast voltage changes which are between 15 to 100 Hertz, because scalp recorded voltages in these frequencies are ranging likely from noise and non-neural sources. So these kinds of signals have to be filtered out. Frequencies between 0.1 Hz and above 18.5 Hz are filtered from the waveforms. They are removed, but also it has a trade-off that filters can distort sometimes the time codes of an ERP waveform and can induce artifactual oscillations when the low cutoff is greater than the approximate 0.5 Hz, or when the high cutoff is less than the approximate 10 Hz. So obviously ERP researchers, when they are analyzing this data, they have to be extremely careful when these filters are being utilized. Filters can obviously be applied to EEG data also, and then they can be applied to averaged ERPs, or sometimes at both steps. Now, how do you compute the average ERP waveform? ERPs are typically small in comparison to the rest of the EEG activity and are usually isolated from the ongoing EEG by a simple averaging procedure. To make this possible, it is necessary to include event codes in the EEG recordings that basically mark when is your stimulus coming. So mark the events that happened at specific times, such as the onset of each stimulus. That is why you will be able to get the best data. These event codes are then used for time locking, to extract segments of the EEG around each event, just before the stimulus presentation and just after the stimulus onset. We have seen in the oddball experiment that the EEG was recorded over a 9-second period in an oddball task with frequent X stimuli that were coming around 80% and infrequent O stimuli that were coming around 20%. Each rectangle, basically the area that you take off, each rectangle highlights a 900 millisecond segment of EEG that begins 100 milliseconds before the event code and extends until 800 milliseconds after the event code to capture processing post-stimulus. The 100 milliseconds before the event code is also used to provide a pre-stimulus baseline period as to what was happening before the stimulus even appeared. Now, these segments of EEG, around 9 seconds of worth of EEG data, are lined up in time. They are chronologically arranged. Stimulus onset time is set at 0. There is quite a bit of variability in the ERP waveforms from trial to trial, and this variability basically reflects the fact that the EEG is the sum of many different sources of electrical activity in the brain, many of which are not which we are interested in, and they are not involved in processing of the stimulus. Now, to extract the activity that is related to stimulus processing from the unrelated EEG, the EEG segments following each of the X's are averaged together into a different waveform, and the EEG segments following each of the O's are averaged together into a different waveform. Any different brain activity that is not time logged to the stimulus will be positive at a given latency on some trials and negative at a different latency on another trials, and if these are averaged together, they sort of go away, they cancel out and they approach 0. So what you are left with is averaged waveforms that are contingent to X's and contingent to O's after your analysis. Now, any brain activity that is consistently elicited by the stimulus across trials with approximately the same voltage at a given latency across trial to trial will be captured, will remain in the average. So, by averaging together many trials of the same type, the brain activity that is consistently time logged to processing this particular stimulus across trials can be extracted from other sources of voltage. So, you can clean rest of the junk away and basically capture this clean signal. Other types of, and this will also include EEG activity that is unrelated to the stimulus and is arising from the non-neural sources of electrical noise. So you have to be very sure of that. Other types of events that can be used as time locking point in the averaging process, for example, button press responses, vocalizations, saccadic eye movements, you can actually work with what kind of events are causing what kind of waveform. The number of trials that must be averaged for each ERP waveform depends upon several factors. For example, the size of the ERP effect that you're looking for, the amplitude of the unrelated EEG activity, the amplitude of non-neural activity, all of that basically, depending on how much noise there is, it will basically decide how many trials you will need to average. So for instance, for a large ERP component such as the P3 wave, very clear results can usually be obtained by averaging around 10 to 50 trials. For smaller ERP components such as the P1 wave, it is usually necessary to average together at least 100 to 500 trials for each type of condition, for each trial type, to be able to see reliable differences between groups and between conditions. Now, another a very important step in this whole thing is the quantification of amplitude. So, what is the amplitude? What is the latency at which that amplitude is peaking? So, the most common way to quantify the magnitude and the timing of a given ERP component is to measure the amplitude and the latency of the peak voltage within the same time window. For example, to measure the peak of the P3 wave, one might define a measurement window, let us say between 400 to 700 milliseconds or a little bit earlier, and find the most positive point in that window. Peak amplitude would be defined as the voltage at this most positive point, and peak latency would be defined as the time of this point. So, as the wave is rising, the peak amplitude will be required as the main amplitude, and the time at which this peak amplitude is reached is known as the peak latency. Finding peaks was the simplest approach to measuring ERPs before the advent of inexpensive computers. Basically, earlier what people used to do was there was a ruler only available to quantify the waveform. So, an approach, sometimes it is also used when you just want to manually look at the data, but it has several drawbacks, and better methods for quantifying ERP amplitudes and latencies have now been developed and are available for researchers. For example, the magnitude of a component can be quantified by measuring the mean voltage over a given time window, and the mean amplitude is usually superior to the peak amplitude as a measure of a component's magnitude. So, for example, once you average the mean amplitude, you basically get a better and more stable measure than just a single peak over a particular trial. Finally, the most important step is the statistical analysis. As in all kinds of experimental methods, in most ERP experiments, an average ERP waveform is constructed at each electrode site for each subject in each condition. So, you basically get subject wise data. The amplitude or the latency of a component of interest is then measured in each of these different waveforms, and these measured values are then entered into statistical analysis just like any other variable, just like you would do reaction times or accuracy. Hence, the statistical analysis of ERP data is often quite similar to the analysis of other traditional behavioral measures. However, ERP experiments provide extremely rich data sets, usually consisting of several gigabytes of data, which can lead to both implicit and explicit use of many statistical comparisons. People basically want to really sieve the data, really beat the data up in a single study, which sometimes can dramatically increase the probability of a type 1 error, because you might be looking for effects where there are none just by arranging the data in several ways. The explicit use of multiple comparison arises, for example, when separate statistical analyses are conducted for each of these several different components. The implicit use of multiple comparisons occurs when researchers first look at the waveforms and then they decide upon the time windows and the electrode sites to basically magnify or quantify this component amplitude and latency. So, if you have your theory beforehand, if you have everything in place, then the probability of these errors is much less and it is reduced. Now, if a time window is chosen such that because the difference between conditions is greatest in that window, then this would bias the result in favor of a statistical significance, because you are already seeing that. Even if the difference may just have been caused by noise. A similar problem would arise if the researcher finds the electrode sites with the largest difference between conditions and then uses those sites for statistical analysis. So, unless your analysis is theory driven, it is based on previous research, there are these two different ways, both of which can lead you to making errors. With enough electrode sites, it is almost always possible to find statistically significant difference between two groups or two conditions at a few electrode sites, just simply due to random noise. So for example, if you have a 128 channel setup or a 256 channel setup, some or the other pairs of electrodes will always be differing with each other, and if you perform enough statistical analysis, some kind of significance will be seen. So that is why it is encouraged to have a prior theory before you get into the analysis, so that you have a sense of what is it exactly that you are looking for, which amplitudes, which latencies, and which sites you are actually interested in analyzing. So one should be very careful with the choice of electrode sites chosen for analysis and the conditions for the comparison. So this is the overall setup, how you make and what are the different components of an ERP experiment. Now we can discuss, while parting, briefly a little bit about the benefits and the disadvantages of the ERPs. For example, the most common advantage of the ERP technique is its high temporal resolution. ERPs provide a continuous measure of processing which begins prior to the stimulus and extends after the response. So it gives you a very good window in time how a given stimulus is getting processed when it is presented to the brain, what kind of process goes on, and how does the brain react after the stimulus has offset. In a behavioral experiment, for example, ERPs give us a measure of the moment by moment activity during the period between the stimulus and response. ERPs actually show us the action in the brain, that is how a given stimulus is processed, and also provide information about brain activity that occurs after a response has occurred, after a feedback stimulus has been presented, which basically reflects the executive processes and determine how the brain will operate on next trial. Say for example, there are phenomena like post-error slowing. You've made an error, the brain reacts in a particular way, it prepares to respond to other trials in a different way. These are all what can be accessed by the ERPs. Determining which process is influenced by an experimental manipulation. Remember, we were looking at the schizophrenia study in the previous lecture. So the ERP methodology, given that it has this continuous temporal information, it allows us to understand the exact cognitive processes that are affected by our experimental manipulations, as opposed to a bunch of other processes as well. Say for example, in a Stroop paradigm, subjects' slowed responses reflect a slowing of perceptual processing or a slowing of response selection process. Remember, in the schizophrenia example, we were able to basically delineate that it was not due to slowed perceptual processing, but basically due to response selection processes. Alright. Also, being able to identify multiple processes that go on when a stimulus is presented, multiple neurocognitive processes. So, ERP recordings actually provide us with a very rich data set that makes it clear that a given experimental manipulation may actually influence not one and not the only one which we are interested in, but several different cognitive processes which may in turn influence several different ERP components. And a given pattern of behavior might be caused by different mechanisms in different experiments, so task-based effects and so on. For example, behavioral studies often treat selective attention as one mechanism, but different manipulations of attention have actually been shown to influence different ERP components. So for example, you might refer to the attentional network phenomena by a fan, and you can see that there are different ways in which attention gets affected. So alerting, orienting, and executive networks are all there, and they might differentially affect the ERP components in the brain. Also, ERPs are able to be, reveal to us covert measures of processing. For example, ERPs can be used to provide an online measure when the particular person is processing the stimulus, when a behavioral response is sometimes impossible or problematic. For example, ERPs can be recorded from infants who are too young to be instructed to make a response. They are also recorded for covert measurement or covert monitoring of people who have some kinds of neurological disorders and they are unable to make proper behavioral responses. So those covert measurements are also possible through ERPs. Finally, ERPs can be actually used as biomarkers in several medical applications, clinical applications, as they can be used to measure aspects of brain function that are impaired in neurological and psychiatric diseases, providing more specific information than an individual patient's brain function that could be obtained by just traditional clinical measures. So a lot of people actually use ERP measures as biomarkers for various diseases. Now the disadvantages. The major disadvantage is that there is, in the ERP signal, there is a lot of information. This is basically the single ERP component, or the EEG waveform represents a sum of many underlying components. It is oftentimes difficult to decompose this mixture of components into individual underlying components. This is called the superposition problem, sorry, and at the same time it is difficult to determine where is the generator of these, where is the natural generating locations of these components. These two problems are the most common impediments for successfully conducting ERP research. Another key limitation of the ERP technique is that a given mental or neural process may sometimes have no ERP signature. So there are obviously dozens of distinct ERP components, but there are also surely hundreds or thousands of distinct cognitive processes that do not have a signature ERP component. So sometimes people may be misled into indicating an ERP component as measure of a particular process. Another limitation that arises from the fact is that ERPs are small relative to the noise level, and many trials are actually required to accurately measure the ERP effect. So that also it takes a lot of effort to get the clean signal out of the ERP waveform, and it basically makes it difficult to conduct experiments with very long intervals between stimuli, and experiments that require sometimes even surprising the participants. Another important thing is that because we are not sure of where the ERP waveforms, where the EEG waves are actually arising from, they have a very poor spatial resolution. You can never be sure unless there is a lot of channels, and now you're using specialized software. It is difficult to be sure of the source of the ERP signal. So because it is a summation of so many activities over the scalp, it is difficult to know exactly where in the brain, which particular points in the brain the ERP signals are arising from. That is why, while ERPs have an extremely good temporal resolution, they have a poor spatial resolution, which make it a poor method to study the space in the brain rather than of studying temporal processing in the brain. Finally, costly. Yes, the setting up an ERP experiment is costly. It takes around $50,000 to more to set up an ERP experiment. So that is also something that is a disadvantage. But once you have a proper setup and you can sort of get on collecting good data, it is an extremely important, extremely useful behavioral method to conduct experiments. So that is all from me in this lecture, and I will see you in the last when we are talking about other topics. Thank you.